So there's a soil microbe, particularly a fungus, that I want to isolate. Came here to find trees that, hi bird, seagull, geese, I don't know. Anyway, so I'm attempting to isolate this particular soil fungus, and it is one of the good ones or beneficial. Good slash beneficial, it depends on the context. So why this one is good slash beneficial is that in its interaction with plants, etc., it doesn't cause disease. What it does, it will help the plant fight against pathogens, so the ones that cause disease. It can help the plant to sequester, so find, absorb nutrients that might not be usually available or that are scarce. It can help the plant increase its growth, etc. So it is, as I said, it is found in many soil, and in isolating soil microbes, whether it's bacteria or fungus, especially the good ones, you want to get the sample from the part of the soil where there are roots, because a lot of these beneficial / good microbes, they are in association with plant roots. And I am going to look for a tree that might look like it's decomposing, or if I can't find a tree like that, then I'll ensure that I take the soil sample from the roots in the area. So that part is known as the rhizosphere. And then I will do my typical technique, or the standard microbiology technique, when you're isolating bacteria or fungus from soil. So I'm going to do the typical serial dilution and plating. And I did that in the Are There Actually 5 Billion Life Cells in my probiotic video? I link it here. So it's going to be that same process. And this particular fungus, it grows green on the agar. So I'm going to use a particular agar that will select for it. So it will prevent other things from growing and allow this particular fungus to grow. But yeah, enjoy the view. I'm enjoying the view. It's a beautiful day. Spring is here, and I'm glad. Okay, oh, the roots of this tree is covered in some sort of ivy. I mean, there are a lot of trees here. Actually, I'm going to include some tree bark. There's a fallen tree here into it, because it can also be found on tree barks. I'm going to look for like whitish discoloration on the tree bark. I don't know, I don't see any, but I'm still going to sample a piece of the tree bark. So I brought my trowel and my falcon tube to collect the sample. I might go over there to find the roots too. I got some soil that has plant roots, some fine roots, and I'm going to get some of the decomposing / rotting tree bark to add to it. and then I'll go enjoy the sun setting. Yep. Yeah, it smells very woody, very earthy. Okay, this is a lot, and all I need is 1 gram. But yeah, the sun is setting beautifully, so I'm going to go check that out. Okay, smell you later. I think that's Flair. Yep, looks like Flair. Flair Airlines. Why did I need to go outside to get soil in order to isolate the fungus that I need? So commonly there are three ways of obtaining microbes, whether it's bacteria or fungus specifically. You could either one order it, so buy it from a culture collection bank. They have one in the Netherlands that we use. You have the ATCC, that's an American one, and then you have the Canadian Fungal Culture Collection, and I think that one is probably just for fungus. Or if you have a friend, aka a prof, whatever, who works at a university and they're using your particular microbe of interest in their lab, you can ask them to share a sample with you, just a little piece, and then you can propagate that, grow that up. Or you can actually isolate it for yourself from environmental samples. So it could be from diseased plants. If you're looking for a pathogen, it can be from the roots, stems, or the soil. So whatever, water, whatever environmental sample. Or from a commercial product, you could buy that and isolate it from a commercial product. So I am isolating it from the soil. Why not go to the usual other two, or the first one where we buy it? I am working on something and I'm just curious to see if I can get it, seeing that that's not the main one we're working with. And so I have my soil samples, and what I'm going to do is I'm going to put the different types that I got it from different places, just to increase my chances of isolating the particular fungal strain that I want. I'm going to put it in a beaker with water that has a stir bar. I'm going to let it stir for about 20 minutes just to get everything uniform. And once that's done, I'll take 1 milliliters or ml and carry out the serial dilution and plating step, which by now I've done it so many times, but it is a common standard way of enumerating microbes. So yes, I'm going to go put my soil in the water, have that stir for 20 minutes, and then come back to do the rest of it. I'll go do some other science stuff in the meanwhile. This is just potting soil, like you can buy from the garden store, Home Depot, or Canadian Tire, I don't know. And this is the one I got from the river. All right, I need help with this one. Oops. Oh, it took the shape of the falcon tube. I have the stir bar in it, so I'll just put it on a magnetic stir and then this will spin. While the soil was spinning for 20 minutes, I prepared the specific agar that I will need to grow the fungus on. So it's water, agar, antibiotic to prevent bacterial growth, and antifungal agent to prevent the growth of all the other fungus that I don't want, but allow the one that I want to grow. That's why it is specific. And I'm going to carry out the serial dilution portion, where I have my original sample
and I'll take 1 ml, so 1 milliliter from the original sample, put it in the first tube that contains water or salt and peptone, shake it up, take 1 ml from the first into the second, then from the second to the third, fourth, fifth, until I get to the sixth. So that's the process of serial dilution.
Many hours later, I had to take not a break, but I had to make up the medium, the agar that I am going to grow or isolate the soil fungus on. And this media isn't the general one that we use or I use for growing fungus. This particular soil fungus, I include some different things in the media so it will select for the particular fungus that I want while preventing ones that I don't want from growing. So I have antibiotic which will prevent bacteria from growing because there's bacteria everywhere as well in the soil too. And then there's also fungus. So I put different chemicals in it that will reduce or slow the growth of other fungus or fungi, plural, while allowing my specific one to outcompete the other one. So I am just going to go ahead and mix each of these. I have decided, because I've done this several times, the very first one is going to be too concentrated, so there'll be too many cells that I can't count. Same with the second one. So I have decided on to add the 10 to the 3, the 10 to the 4, and the 10 to the 5 dilution. So countable range between 20 to 200 or 30 to 300 colony forming units or cell of the particular fungus that I want should be present between the 10 to the 3 to the 10 to the 5 range that I can count. So you'll see me do that now.
Reminder, already completed the serial dilution part. So I'm going to select the 3 dilutions that I'm going to use, the 10 to the 3, 10 to the 4, and 10 to the 5. I'll take 0.1 ml or 100 microliters from each plate, from each tube, and put them on 3 separate plates. So I should have 9 plates at the end. I'll use a sterile glass rod or hockey stick to rub or spread the liquid from the different dilution series onto the surface of the plates and let them dry, and that is why it's called serial dilution and plating.
These are the plates with the 3 dilutions that I use, the 10 to the 3, the 10 to the 4, and the 10 to the 5. And as you can see, the plates are devoid of cells, but we'll come back and see in a few days if there's growth, and especially the growth that we want. So the growth that I want, they should grow green. And then as they get bigger, they should form concentric circles, but I'm looking for green growth. And once I have green cells, then I'll take a little piece of it and streak it onto a new empty media of the same type. But yeah, this is the incubator that I'm going to put it in. I might change the temperature, but 25 is usually good. It's like a little oven. And I'm going to leave it in the dark so I won't turn the light on. Put them on the same shelf, just in case there are differences between the 2 shelves in terms of the temperature, etc. But yeah, as Myra Carey would say, festive. Something that I love when the boats, whether it's a logging or just recreational boats, go by. Oh, there are no logs here, but usually there are logs, and then the wave just pushes it to the shore. Is it going to dissipate before it gets here? Do you see the sinus? I should remember this, but I don't. I learned it in physics. Okay, it might crash against the rocks. Wow. The big swell of the wave. I love it.